Okay, yes, thank you very much. And I would also like to, well, obviously thank you for the invitation, but also thank you for suggesting this topic of my talk, which is unlocking the quantum design of photosynthesis for bio-inspired solar energies. This is a topic that lies very close to my heart, and in this talk, I will connect photosynthesis, so biological systems, with solar technologies, and obviously everything is based on quantum science, quantum technology. Okay, so the connection between photosynthesis and solar technology is quite straightforward. If we just think about what photosynthetic systems are, they are absorbing light from the sun and then they convert this light into useful forms of energy. The connection to the quantum science or quantum technology might be a bit more difficult to see, but that is the whole purpose of this talk. Okay, so before we dive in there, I just want to first show you the magnificent scale of energy conversion that we are dealing with here. So photosynthesis is dealing with energy conversion, and we're talking about 105 billion tons of biomass that is produced annually by photosynthesis. Okay, so this is equivalent to 2 Great Pyramids of Giza per hour, every hour. So this is really energy conversion at a massive scale, and half of this happens in water. So there's a whole gallery of photosynthetic organisms, beautiful designs, many different types of designs, and so much that we can learn from them. So photosynthesis, because we're dealing with natural solar cells, they are actually a huge source of inspiration for solar technologies. And I'm just going to give you a glimpse of that. Let's first have a look at this equation, which I'm sure all of you have encountered already at school. It is actually a very deceptive equation because here it is written as a very simple equation. And what is here in square brackets, you might be used to that being written as glucose, so one type of stable product that's formed. It's deceptively simple because this single equation actually comprises more than 170 different chemical reactions that are taking place. Let me rewrite this equation in a slightly different form. Let me write it like this. We are dealing with cheap molecules, CO2 and water. Why are they cheap? It's because they are extremely abundant, and then light is used as a source of energy just to initiate the process, and then the cheap molecules are combined into fuels, stable molecules. So it is actually fantastic to think how photosynthetic organisms do this, to use cheap, very abundant materials or molecules in order to generate fuels. And we actually envy an equation such as this. There's globally big research programs trying to mimic this process of using abundant, cheap materials in order to produce fuels. And we have not even come close to this combination of low cost and efficiency. So even in this respect, there's a great deal that we can learn from photosynthetic organisms. If we are thinking about fuels, so what type of fuels do we have here? Well, the end products of photosynthesis are carbohydrates, okay, which can quite easily be transformed into liquid fuels like the petrol, diesel that we are using in our vehicles. But if we are interested in other forms of energy, we can actually chop the process off earlier, and we can use electrons. Well, electrons are actually transported in this whole photosynthetic process through a very long chain of molecules. So these electrons can be extracted, and free electrons are then electricity. Or we can also use the protons that are generated to form hydrogen. Hydrogen is also it's a green fuel, a green form of energy. So there's a lot that can be said about this, which is not the focus of this talk. I just wanted to focus on the fact that there are many different types of fuels, forms of energy that for which we can use the photosynthetic process as a great source of inspiration. So if you're interested in this, the different forms of energy products that can be produced, I've given a mini school lecture on a YouTube channel of NIFEX with a link here, which you are most welcome to visit, and giving a bit more information there. Since this is a discussion forum on quantum science and technology, you might be wondering by now, where does quantum actually come in? It's right there where we have light. So photons. This process happens one photon at a time. Okay, so exactly here where photons come into the equation, that is where quantum mechanics takes place. And it's not only simple, or relatively simple quantum mechanics of normal photon absorption and the generation of electronic excited states, but there's much, much more that is happening there. So let me give you a glimpse of that. Okay, let's first have a look at the so-called natural solar cell. So the very first step of photosynthesis happening on time scale of femtoseconds to a few tens of picoseconds. That's where the fascinating quantum mechanics comes into the picture. Okay, so what happens? A photon from the sun is absorbed, and this produces an excited state. An electron will be transferred to a higher energy state, and that excited state is then channeled down amongst a lot of other systems towards the so-called reaction center. Okay. So this whole extended system, which comprises many different protein complexes, pigments like chlorophylls that are attached to it, they form the photosystem. It's an extended network, and we often call this an antenna. So it's an extended antenna that is responsible for capturing the solar light and then funneling the energy to the reaction center, where the first steps of energy conversion take place. Okay, so this idea is conceptually similar to what might be more familiar to you. In this case, I'm showing you a solar energy concentrator. So conceptually, it's similar. You have all these different antennas, and they reflect the light to the receiver, and that is where you want to have the energy, because that's where it will be converted into more stable forms of energy. Okay, what is the role of quantum here? So one great aspect is that in the natural system, this whole process, from the absorption of photon until it reaches the reaction center and a charge gradient, actually two types of charge gradients are formed to temporarily store the energy. This whole process happens with a 100% quantum efficiency, or basically 100%. Okay, and that means that the energy of each absorbed photon is used for photosynthesis, and that energy is eventually stored in more stable forms of energy. Okay, so yeah, this is already fascinating to think about that. So what is the key to reaching this very high quantum efficiency? What is the trick? It is the extraordinary high pigment density. Okay, let me show you an example from the main light harvesting complexes of plants. Okay, so let's just take a plant leaf and we zoom into it. Here's a light microscope image. You can see the cells with the different chloroplasts. If you zoom into the chloroplast, now we are using an electron microscope, you can see folded membranes, and it's inside these membranes, like membranes are like soap bubbles. Inside these folded membranes, that's where photosynthesis takes place. Here's an example of photosystem 2, and then one antenna system of this is called light harvesting complex 2, the main light harvesting complex of plants. And its dimensions are, its shape is, it's like a disc with a diameter of about 7 nanometers, thickness of about 4 nanometers. And now imagine packing 54 pigments into this small space. The types of pigments that we are dealing with here are chlorophylls, where just this ring of the chlorophyll has a diameter of about 1 nanometer, and carotenoids, okay, which have a length of about 3 nanometers or even greater. So imagine these relatively big molecules, 54 of them packed into this small space. Then you get really a high density of pigments. So the center-to-center distances between the pigments ranges between 1 and 2 nanometers, sometimes even a bit less than 1 nanometer. That is a very high density, and that is responsible for forming very interesting quantum states. Okay, we call these quantum states molecular excitons. Okay, so it is a quantum coherence, quantum superposition that is formed, which we can write like this. So we are now not talking about individual chlorophylls anymore, but we must consider a quantum superposition. So in other words, the system samples all possible quantum states with some probability, and we must describe all the possible states at the same time. And one of the consequences of this process of forming the molecular excitons is that the different energy levels are split. Okay, and because of this and the fact that you have this quantum superposition, there's a whole range of benefits, biological benefits that are formed. The first one being more light that can be absorbed. When the light is absorbed, the energy is funneled much more quickly to the reaction center. Energy transfer is much more enhanced. And the excitation is also not lost. So once you have this absorption, this energy is going to be used for photosynthesis. So there's going to be much less trapping and energy loss of the energy inside the antennas. And lastly, there's also faster charge separation in the reaction center. So another step that ensures that the absorbed energy is going to be used for photosynthesis. Let me give you a few examples of some of these benefits that are created. So firstly, because of this energy splitting, so if a range of pigments are very closely coupled, you will get an energy ladder. So the energies are all split into what we call an energy manifold, and that is responsible firstly for enhancing the light absorption. Let me show you an example from purple bacteria. So that is a type of bacteria that lives in water, and the main light harvesting complex has this beautiful concentric symmetry of the different pigments and proteins that are used. If we take away the protein, this is what remains. So 2 concentric rings of pigments. So very similar to the chlorophylls that we get in plants. Here in this bottom ring is a large number of pigments, and they are so closely spaced that they form these very strongly coherently coupled systems. And they are responsible for this energy ladder. So the excitation energy levels are then split into a whole range of different energies. And if you just put them side by side, okay, here we have the wavelength, which is equivalent to energy. The lowest state will absorb energy that is much lower than the highest state. So in this way, many more photons of many different colors can be absorbed just because of this energy splitting that is created by this electronic coherence. Okay, yeah, so that mainly explains the enhanced light absorption that we get. Let me also give you an example of the enhanced energy funneling. And for this, we are looking at, let's just conceptually look at a system like this. And this actually portrays one light harvesting complex that is very commonly investigated, where you have an input and an output position. So energy is going to be funneled from other systems towards this site. Okay, so that is the input, and then you want this energy to reach this output state. Okay, so now if we consider all these pigments to be separate entities, okay, say that we start there, how many steps of energy transfer do you need in order to get to the final state? Okay, so we are starting there at the input, and then we need to go down this energy gradient, okay, 1 step at a time. Okay, so here we have quite a number of steps, and that takes a long time to reach this final state. But if we now form these delocalized exciton states, they are spatially delocalized. So you will have that, for example, these 3 pigments are coupled together, these 3 are coupled together, these 3 are coupled together. And now if you excite this pigment, it's as if you're exciting this whole state. Okay, so if we have this input, we now only need 3 steps until you reach the output state, which is obviously much faster in terms of energy transfer down this energy gradient. Okay, let's have a look at this 4th physiological benefit, less trapping and energy loss in antennas. Okay, so once again, since we have this delocalized state, so quantum coherent state, we're not talking about the individual pigments anymore, but quantum superposition of all of them, and the spatial extent is also much greater. Okay, so imagine trying to play soccer with a tennis ball on a rough grass field. What is going to happen? Let's say it's a grass field like this. If you're going to kick this tennis ball on this rough grass field, it's not going to get very far because it's going to get stuck in a dent just every few meters. The reason for that is that the disordered energy landscape, well, let me say the landscape of the grass field has the same dimensions as that of the tennis ball. Okay, so you will get something like this. Okay, you're starting there. Okay, that's the input. This is equivalent to absorbing a photon by an antenna system, and you want to get the photon here in the reaction center so that the charge separated state can happen. Okay, but now you're kicking the tennis ball and it gets stuck in a dent, and then you kick it again and you have to get it out of the dent, kick it again. So it takes a long time, and for an excitation, there's a very high probability that you're going to lose this energy because this excited state doesn't live very long. Okay, you only have a few nanoseconds and then this excitation is lost. So that's a pretty short time. Okay, so this is not going to work. But if we deal with a delocalized excitation, then it's equivalent to having a soccer ball, which has a much bigger size than the roughness of your grass, the landscape. Okay, so now if you're going to kick this soccer ball, it's going to roll smoothly over this landscape, and then in the equivalent case of this excited state, or this delocalized exciton, it's very rapidly going to be channeled towards the reaction center and then be trapped exactly at the place where you want it to be. Right. So let me show you 2 examples of how photosynthetic systems use this concept and actually by this tune their quantum nature. Okay, so the first system is that of diatoms. The main light obviously consists of diatoms. Diatoms are a major type of algae in the ocean with a lot of similarities to plants when it comes to their light harvesting or their photosynthetic system. Okay, so their light harvesting efficiency is extremely sensitive to the distance between the pigments and the relative orientations between the pigments, which are mostly chlorophylls, again, in the light harvesting complexes. And all these pigments are embedded in a protein which acts as a finely tuned matrix to put the pigments in precise positions and orientations. And this is really a fascinating design feature because even a small change in the positions or relative orientations can distort the quantum superposition, which I've explained to you, and as a result, compromise their light harvesting efficiency. So it's the exact positions and orientations are very important. So if we now zoom into the main light harvesting complex and in the center, okay, where an excitation energy is going to be funneled, which is going to stay until it will be transferred to a neighboring complex and eventually to the reaction center. There in the middle, we have 3 chlorophylls that are very closely spaced. So they form a very strong excitonically coherently coupled system. 2 of these chlorophylls are chlorophyll A, but one has a different identity. It's a chlorophyll C. Because of this fact, it's distorting this quantum state completely to such an extent that it is actually reducing the light harvesting efficiency. But more than that, this protein is designed in such a way that it can actually switch between different states. It can switch the light harvesting efficiency by just slightly changing the structure. And when it changes its structure, the pigment positions and orientations are slightly altered, and that will then completely change their excitonic coupling. So very small changes can have very large consequences. Okay, and another example is the main light harvesting complex of cyanobacteria. And this actually presents us with an even more fascinating example. This is an enormous system. If you just think about atoms and molecules, how small they are. So here we are talking about a diameter of 70 nanometers. This is enormous for a biological pigment protein complex. Okay, so as I explained, the light harvesting efficiency is extremely sensitive to the distances between the pigments and their relative orientations. And we have put this light harvesting complex inside a focused laser beam, and then we performed single molecule spectroscopy. So this is a technique by which you can observe the emitted light from one protein complex at a time. And when doing that, what we saw was that the photons that they emitted, so the photon stream, as you continuously excite them, it switches on and off. This is really fabulous that they are switching this on and off. So it's just a constant excitation intensity, but then the emitted photon stream switched on and off. And what happens is that this protein, again, changes its structure slightly, and by just this slight change, the relative pigment positions and orientations are slightly altered, and these small changes have a huge effect on the electronic couplings and eventually to their light harvesting efficiencies. Okay, so how can this knowledge be applied to organic solar cells? Okay, let's consider two organic polymers. Okay, let's say that we're considering these two polymers, and these are the two arrangements of them. And just as with biological systems, the relative positions and orientations of the individual molecules are again crucial. So we have to differentiate between different types of couplings. So let's say these are the different dipoles. So dipole-dipole coupling, this is what determines the different types of interactions that we are dealing with. Okay, and here we have intramolecular interactions and intermolecular interactions. And if they are spaced close enough, they also form molecular excitons, so quantum coherences, with these energy levels being split and the excitation being delocalized across big parts of the molecules. Okay, and there are two types of pigment aggregates that we are dealing with, so-called J aggregates when you have this head-to-tail arrangement, and H aggregates when you have this sandwich arrangement. Okay, so here's an example of that. The stacking of the different polymers forming the H aggregates, and then the intramolecular couplings that is then forming a J aggregate. Right, so J aggregates are better at generating excitons, which will optimize the current that is generated. But H aggregates are again better at exciton dissociation, and that gives us more useful voltage. Okay, so the exciton generation, exciton dissociation are often competing aspects, but we need both in the case of a solar cell. We need a high current and we need a high voltage, because current times voltage gives you the power output of your device. Okay, so how can both of them be optimized? Well, once again, we can look at photosynthetic systems, and then especially self-assembled systems, because for these organic systems, we want to be able to engineer how self-assembled organic aggregates are packed, how the pigment packing, the molecular packing looks, and that is extremely difficult to predict. Okay. But once again, for the photosynthetic systems, we can gain a lot of inspiration. So in this case, let's have a look at the green sulfur bacterium. And it's a bacterium that lives at very deep levels below the surface of the ocean. And so the light harvesting system in this case, doesn't contain proteins, but just self-assembled pigments, bacterial chlorophylls, which are very similar to chlorophylls. And they form a very specific mesoscopic structure and also molecular structures. On the molecular level, you have perfect molecular crystals. Okay, so these molecules will be arranged, will be stacked like face to face in a very, very specific way. Okay, so you have here very precise H aggregates and J aggregates. Basically, these systems are just exploiting the properties of J aggregates and H aggregates together in a fascinating way. Okay, just a last couple of things I would like to say. So I would like to broaden what we can learn from photosynthetic systems, not only to the application of solar energy, but also more broadly when we think about quantum technologies, because it's very interesting that many of the key enabling technologies that the EU is seeking to develop are based on quantum mechanical phenomena, and they find examples in nature. So in other words, mankind is desperately seeking technological solutions which already exist in nature for many of these technologies. And here are a few examples from the fields of photonics, nanotechnology, nanoelectronics, advanced materials, and photovoltaics. And here are examples that I've shown during this talk: light harvesting complexes, photosynthetic systems, how they are exemplary systems for some of the technologies that we wish to develop. Okay, another aspect is, let me introduce to you briefly another quantum state, and that's called a charge transfer state, which happens when you have a very strong overlap of the electronic orbitals of two closely spaced molecules, such that you get a strong charge gradient. So then your electron cloud is shifted much more towards one molecule than the other one. Now, photosynthetic systems exploit this type of state for different purposes. And here is one example of that. Study which I did back in my PhD. And I looked at one of the light harvesting complexes in photosystem 1 of plants, and then specifically this light harvesting system. Okay, and if you zoom into this system, there are two chlorophylls that are again very closely spaced. They are so closely spaced that they form this charge transfer state. Okay, but and this charge transfer state overlaps with an excitonic state, and this overlap can be switched on and off by changing the distance between these two chlorophyll rings by less than 1 angstrom. Okay, so an angstrom is 1/10 of a nanometer. It's a very small distance by which you can switch this coupling on and off. And we investigated this spectroscopically, so you see a very strong redshift, and we can explain all of this very nicely quantum mechanically. But this idea is actually very interesting, that such an extremely small change can be sensed. Well, we can sense it spectroscopically. Okay, and this system is also using it for a functional advantage. Okay, so if you're talking about such small changes in distances, this is actually a very sensitive strain sensor. Just one example of how we can get inspiration from nature. Okay, so I would like to end with this, a few take-home messages, what I wanted to portray from this talk. So firstly, that nature is a rich source of inspiration, even for quantum technologies. Photosynthetic light harvesting complexes exploit and finally control quantum phenomena to enhance their biological function in several ways. And a few examples I've shown are that super efficient organic solar cells can be created in principle through programmable self-assembled molecules. Secondly, highly sensitive strain sensors can be created based on some examples that we find in photosynthetic systems. And if we think about the fact that these systems are single photon emitters, so that's something that I have not touched on yet, but something that we are currently investigating in our lab. So biological single photon emitters have big applications in biotech applications, quantum biotechnology applications, where you want to have a reliable single photon source. So one main question is, can these biological single photon emitters be tuned to deliver on-demand photons for biotechnological applications? Okay, one last slide, food for thought. So yeah, that's one reason why I'm very interested in the overlap between biology and technology. And obviously, it's a lot of physics that also comes into the mix. And then finally, people who have contributed, the current research group with 2 visitors there, all the different funding agencies. You can find our group on social media. We're quite active there.